Hello students, welcome to the SRBPS virtual classroom program. I am your biology educator Prerna Rathi.
So students, now let us start the last part of this chapter, that is the endomembrane system. In this we are going to learn about various organelles which are a part of the endomembrane system which together work in a coordinated manner. So basically they divide the cell into compartments. What are these compartments? The endoplasmic reticulum, the Golgi apparatus, the lysosomes and the vacuoles. They are responsible for production of various macromolecules like proteins and lipids in the cell. And as you can see in this particular diagram children, ये सब ना बिल्कुल साथ में कोऑर्डिनेटेड फंक्शन परफॉर्म करते हैं. ठीक है, ये देखिए रफ ईआर से प्रोटीन बनकर आया, यहाँ से लिपिड बनकर आया, इसके फॉर्मिंग फेज से एंटर हुआ, मैच्योरिंग फेज से बाहर निकला, पैकेजिंग होकर निकल गई, and further lysosomes are formed and it is stored in vacuoles. So they always perform a coordinated function. That is why they are a part of a system known as the endomembrane system. Let us now understand the concept of ribosomes. What are ribosomes? Ribosomes are children. They are organelles which contain protein and ribosomal RNA. They are made up of ribosomal RNA or ribonucleoprotein. There are free ribosomes and bound ribosomes. Free ribosomes are present in the mitochondria, the plastids, and as well as the cytoplasm also, and the bound are present on the surface of the rough endoplasmic reticulum. Each ribosome is always made up of two units, the large subunit and the small subunit. There are two different types of ribosomes in the prokaryotes and the eukaryotes. The eukaryotes have the 70S ribosome and the eukaryotes have 80S, prokaryotes have 70S. The prokaryotic ribosomes are made up of 50 plus 30S units and 60 plus 40 in case of eukaryotes. Here S stands for Swedberg unit, which is the sedimentation coefficient. So sedimentation coefficient क्या बताता है? जब हम इस मिक्सचर को सेंट्रीफ्यूज करेंगे तो ये कितने टाइम में, कितने रोटेशंस, कितने स्पीड पे वो सेग्रीगेट हो जाएगा. दैट इज द सेडिमेंटेशन कोएफिशिएंट. Okay, what is the function of ribosomes? They help in translation process, converting DNA information into protein. So they are useful here, especially the mRNA. Can you see here in this diagram? This mRNA is trapped between the ribosomes and the code over it is read in order to convert it into a polypeptide chain which will lead to the formation of protein further. Then we have to discuss centrosomes. What are centrosomes? It is an organelle which is made up of two mutually perpendicular structures known as centrioles. So each centrosome is made up of two centrioles and each centriole is made up of 9 equally spaced. Can you see here? They are made up of 9 equally spaced peripheral fibril or tubulin proteins. This ABC, this is marked, this is each of this is a microtubular tubulin protein. The center is known as hub and there are these triplets are present. Okay, triplets of fibrils are present in the periphery. Okay children. Next, we have to learn about the structure of cilia and flagella. It is very important. So there is a plasma membrane and instead of triplets, there are doublets of peripheral microtubules. उसमें 3 थे, यहाँ पर कितने हो जाएंगे? 2 हो जाएंगे. इनके बीच में, if you can see the small light green color, these are interdoublet bridges. 2 doublets को combine करने वाले bridges होते हैं. ये बीच में जो 2 होते हैं, these are called central microtubules, and the spokes which connect the peripheral tubules to the central sheath are known as radial spokes. This structure, students, is known as the 9+2 arrangement. This arrangement is known as the 9+2. 9 doublets and 2 these. Okay, so this is 9+2 arrangement. Let us now discuss vacuoles. Vacuoles are empty spaces. They are like empty blank spaces which would be used for storage, especially in case of plant cells which have large central vacuoles, where the vacuoles are responsible for osmoregulation as well as storage of various materials. The food, the waste, everything extra is stored in the vacuoles. Also the membrane of the vacuole, can you see here? Okay, the membrane of the vacuole known as tonoplast here is also responsible for, this tonoplast is responsible for regulating the osmosis in the plant cell. Understood, children? The last we have the cytoplasmic inclusion. Cytoplasmic inclusions क्या होता है? Small storage structures होते हैं. These are diverse intracellular non-living substances. They're just a collection of substances. They can be stored foods like carbohydrate, fats, proteins. ये store हो सकते हैं. कुछ pigments store हो सकते हैं. कुछ granules हो सकते हैं. ठीक है? These structures were found by O.F. Mueller in the year 1786. Lastly, we have the difference between plant cell and animal cell. Everybody knows plant cell has a cell wall, animal cell doesn't have. Animal cell has many small vacuoles, plant cell has one centrally placed large vacuole, chloroplast, absent in animal cell, present in plant cell. The shape is variable in case of animal cell, whereas plant cells have a regular shape because of the presence of the cell wall. The centrioles, a pair of centrioles are present in the animal cells, and centrioles are absent in case of plant cells. Okay, next we have the difference between prokaryotic and eukaryotic cells. Prokaryotic, as the name suggests, pro means pehle, karyon means nucleus. The cells, the primitive cells which do not have a proper nucleus are called prokaryotic cells, and which have proper true, u means true nucleus, they are called eukaryotic cells. Prokaryotes have naked DNA which is not bound by any protein. DNA is normally circular and it has no non-coding regions. All of the DNA is coding. Eukaryotes is opposite. No nucleus is present in prokaryotes. No membrane-bound organelles are present. 70S ribosomes are there. It reproduces by binary fission. Single chromosome is present. And normally they are smaller in size. Here I have given you the diagram of an animal cell and a bacteria, which is a prokaryote. Okay, so there is a diagram of a prokaryote and eukaryote, both are there. I hope you have understood all the concepts of this chapter. Next we will be dealing with the cell division chapter after this.
I hope you all understood what I taught. I suggest you all make complete use of the YouTube channel to enhance your learning. So until next time, goodbye, take care.